Dear guests, our university hosts today a multi-annual scientific event, on the one hand prepared by the teaching staff and students in Iași University of Life Sciences, and on the other hand by our colleagues from the kindred universities in Romania, from Bucharest, Cluj, Timișoara, as well as by our partners abroad. A wide range of topical research papers will be presented both in plenary and by section at faculties today and tomorrow. Please accept all my gratitude for your presence here, and we now invite to the stage our rector, Professor 𝗟𝗶𝗳𝗏, for his welcome speech in the Congress opening ceremony.
Dear guests, dear colleagues, students, ladies and gentlemen, I would like to express my pleasure to welcoming you to the opening ceremony of the 12th International Scientific Congress, Life Sciences Today for Tomorrow. This Congress is organized by Iași University of Life Sciences under the patronage of the Ministry of Education and Romanian Academy for Agricultural and Forestry Sciences. It is the most important scientific event of our academic year, with over 330 people registered from the European research area, from outside Europe, as well as from other continents. All these people are coming from universities, research stations, and companies, being here to bring the scientific news into the attention of the participants. Referring to the participants, I want to highlight the presence of our colleagues from Justus von Liebig University of Gießen, Germany, with whom we celebrate 47 years of good collaboration, started by Professor Constantin Vasilică from Iași and Professor Eduard von Boguslavski, who is Doctor honoris causa of our university from Gießen. And also we celebrate 30 years since we signed the first official agreement after 1990. Today, 278 scientific works will be presented live in extenso, in plenary or in 13 dedicated sections, complemented by 5 workshops and 2 book launches. I would like to thank all the participants in this scientific event, senior researchers, young researchers, PhD students, as well as graduate and undergraduate students who begin their journey on this unbounded path of knowledge for being here. Life sciences and agriculture have today a very important role in the global economy and an important development prospects in the European context due to the very good pedoclimatic conditions and the big potential of organic production. But in the current global context, agriculture must be adapted to climate changes by applying new conservative technologies, including smart agriculture and artificial intelligence. The challenge is to use modern research methods to ensure the improvement of the level of agricultural productivity without any damages for the environment, represented by water, air, soil quality, and biodiversity. Another challenge is food security, with organic farming on the upward trend, growing from 1.4% of the total agricultural area 10 years ago till 42% at the end of this decade. Research also means increase of food quality, with new technologies leading to a circular economy, where nutrients and organic components of the soil have a central role. Last but not least, the livestock sector and animal pathology are the keys to ensure the biosecurity of food in the farm-to-fork circuit. During the Congress, I invite you to discover the news regarding the research in all these fields, together with the specialists who agreed to present their scientific papers and are ready for discussions aimed to ensuring a healthy life in a healthy environment. Also, we are looking forward to the presentations of main speakers with comprehensive topics and new European approach on the number of challenges of common agriculture policies whose pillars mark the future of the research in the field. And finally, I hope that this Congress will be a good occasion for everyone to meet interesting people, to make new friends, and to find ideas for new common projects. I wish you to have very interesting meetings, a pleasant stay within the University of Life Sciences and also in IASI, and I declare open the proceedings of the Congress. Good luck, everyone.
Dear guests, as you saw in our program, we start plenary sessions. So the first paper presenting by Dr. 𝗂𝗏𝗄𝗧, it's Dissecting Qualitative and Quantitative Resistance Against Blackleg in Oilseed Rape. Please, Christian.
Thank you very much for the invitation. Okay. I think this is about the 5th or 6th time I'm here. I enjoyed it very much all of the time. Before I start, sorry. I want to show some pictures from the past, which were just mentioned. 47 years of signing an agreement with collaboration. Some of the people here which were instrumental in this, Professor von Boguslawski in Gießen University, and here we have Professor Vasilika on the left. And in 1993, they renewed this collaboration. So in 3 years, we have to celebrate 50 years of collaboration, and a lot of students have been going back and forth between our two universities. So I want to talk about plant disease today and about dissecting qualitative and quantitative disease resistance. So very often these two types of resistances are considered very different, but actually we found out that it's not so clear. And this sometimes is difficult to dissect in the field. This is a disease I'm talking about, blackleg or stem canker of oilseed rape, caused by a fungal pathogen, Leptosphaera maculans, and the asexual form is called Phroma lingam. In Germany, there are a lot of varieties registered, available to the market, which have resistance against this fungal disease. This is a qualitative type of resistance. So that means it's race specific. It doesn't work against all races in all fields. And the newest one is called 𝗂𝗲. So 29 cultivars in this list have this resistance the farmers can choose from. However, this resistance is often broken very easily in the field. After 2 or 3 years, the resistance is not functional anymore. And that's a big problem. So breeders now try to introduce other 𝗂𝗲 genes, for example, 𝗂𝗲S. This is working based on the gene-for-gene hypothesis, which was introduced a long time ago by 𝗀𝗏. So this is a fundamental idea in plant science, that each resistance genes interact with one gene of the fungus or the proteins. And we have the same with the 𝗂𝗲 genes here. Here's an example. We have on the plant side a resistance gene, 𝗂𝗲1, and we have in the fungus another gene which is called an avirulence gene, AVRLM1, and only if they can bind, they are recognized, the plant is recognizing this protein of the fungus, we have resistance. But what's happening in the field is that the fungus is mutating very fast. And then this fungal effector changes, that is depicted here by a small a, AVRLM1, starting with a small a mutated form, and this is not recognized by the plant anymore, and then the resistance breaks down. And what we can do as breeders is to introduce new R genes. So we always have to go in the cycle and find new R genes and introduce them. For example, the newest one, 𝗂𝗲7, which is very common now. So now the plant has 2 𝗂𝗲 genes. The fungus has 2 avirulence. One is functional, the other is not functional anymore, and the plant is resistant. And of course, this can again mutate, and that's what's happening in our fields at the moment. So we have to find better ways to create durable resistance. How could we do this? So one strategy of the breeders is to stack all these genes together and to rotate them so the resistance is not broken so easily in the field by mutation of the pathogen. Another strategy of the breeders is to combine it with quantitative resistance, so with many thousands of minor effect genes. Now, how do we get these quantitative resistance? This is difficult in the field because there are all these races of different avirulence pathogens in the field, and the small effect quantitative resistance in the field is masked. We cannot detect it easily. If we don't know exactly which fungal pathogens are in the field and also which R genes our genotypes have we put in the field. So what is happening here, you see on the left, you grow a susceptible cultivar the first year. There's one red dot. This is a mutated spore of the fungus, and it's minor. It doesn't make much damage. But if you grow a resistant one, the plant looks fine in the first year, but this increases. So you are selecting for the increase of this mutated race, and then you have 3 dots. And the next year, all the other ones cannot survive on the plant. You have even more. And after the 4th year, your plant is not resistant anymore. The resistance is broken. And this we have to try to stop. So one is management of R genes. So give the farmers 3 different cultivars with 3 different R genes. Every year you grow a different one, and then the resistance is more durable. Or to find quantitative resistance. This shows where the resistances are broken in Germany. So the way to read this is these are 7 locations in Germany, 7 fields, and 100% means the resistance is lost. 100% of all isolates we took from this field were not recognized by the plant anymore. The plants were susceptible. So you can see 𝗂𝗲7 has about 17% in Germany. 17% of isolates already break this resistance. And in 7 or 8 years ago, it was 1%. So it's going up, and we have to do something about it. 𝗂𝗲9 is not working, 𝗂𝗲4, 𝗂𝗲3, 𝗂𝗲2, they're all in some of the plants, but they're not working anymore. So we have this qualitative resistance. The quantitative resistance has minor effects, but we know that if we combine it, resistance doesn't break down after 3 years. It has been shown it holds at least 8 years. It has minor effect, it's broad spectrum, it covers all races, that's an advantage. We need molecular markers, but that's not a problem nowadays anymore, we have thousands of markers. We cannot measure this in the field easily because we have all these different races with different avirulence gene in the field. We have to get a specific isolate and do this in the greenhouse. And that's what we're doing here. We look for adult plant resistant, quantitative resistance. This shows the inoculation and the disease growing. We measure volume of diseased tissue, but we also have to make sure that all our genotypes or the population doesn't contain any qualitative resistance. For this, we screened 644 field isolates and we found one, isolate 1415, which has all mutated A virulence gene. So it's all small a, what you see here. And this one creates these lesions on all the plants in our population, so meaning no qualitative resistance. If we measure anything, it should be quantitative then. And that's what we did in this population. We did adult plant screening for quantitative resistance. This is a genotyping, so we have this SNP array, which covers 60,000 probes to measure the genetic composition of every single plant. And we also did use Oxford nanopores, so the newest long-read technology to sequence the genomes of the parents to find out if any polymorphism we find could be, we could identify the genes which are responsible in there. So to our surprise, what we found this here shows on the x-axis the chromosomes. Every dot is one marker measured in 360 genotypes. So genotyped in these and the phenotype data for this. If this on the y-axis is higher than a certain threshold, this position and this molecular marker is significantly associated with resistance. So we found two regions, and one contains RLM9. That was surprising for us because we excluded that all these R genes have an effect. We excluded qualitative resistance by the setup of our experiment. So what's going on here? Is this a region where more than one gene is, one involved in qualitative resistance and others involved in quantitative, so a cluster of genes, or does this gene have a function on both, on qualitative and quantitative resistance? So when we sequenced the parents, we found out there was, in some of the parents, a large 6,000 base pair insertion in one of the introns. And this gene codes for a wall-associated kinase-like protein. Here you see 3 of the parents on the left look like this, and 4 of the parents on the right have this big insertion with transposons inserted. And what is more important is that in one of the domains, there are stop codons disrupting this kinase domain. So maybe quantitative resistance is triggered by this RLM9 gene, and if you have a stop codon in there, it's totally lost, the quantitative resistance. And you can see this if you test all the offspring. So in yellow, the ones which have no insertion, in red, the ones which have an insertion and have stop codons. And in the 6 different subpopulations, you see that the ones with red have a higher susceptibility on average and statistically significant. The ones Lorentz in the middle, where there's only one yellow bar, this is because the 2 parents both have no insertion, and if you cross them, it's not segregating. It's known that now, just 2 years ago, it was figured out that RLM9, RLM4, and 7 are actually alleles of the same gene. So that's the same gene, the same position, just different variants of this gene. So I put them in a phylogenetic tree here, and you can see that RLM4 and 7 at the top, which provide qualitative resistance, also have the 6 kb insertion, the red plus, but they trigger qualitative resistance, race-specific resistance. And on the bottom, the ones from our study in red with an insertion, but with a stop codon. There's a loss of quantitative resistance. And the yellow ones have quantitative resistance. So maybe these stop codons are responsible for quantitative resistance. So what are these wall-associated kinase-like genes. They are well known. They are involved in cell expansion and growth. And usually, this is a normal procedure in the plant. When the plant grows, then pectin methyl esterases are degrading parts of the cell wall components, and they are incorporated somewhere else. But also it's known that this wall-associated kinases will react to damage-associated molecules created by pathogen digestion of the cell wall. They are called here OG, oligogalacturonides, and so there's an overlap of the whole thing with resistance and response to pathogen, but a very general one. They will react to many different fungi, not race-specific. So from the description of the protein, it already looks like this is not just qualitative resistance, very specific, but it's broad spectrum. And if you look with a comparison of, we have 100 of these genes in rapeseed and 26 genes in the genome of 𝗂𝗶𝗳𝗏. The most similar one to RLM9 is VACL10, and this is involved in broad range resistance, so not race-specific. So to summarize, we have here a gene, RLM9, which might be involved in both qualitative and quantitative resistance. And there's a kinase domain, which might be responsible. These genes have been found in many different crops, maize and rice, and against many different diseases, but often they are involved in quantitative resistance. And so this is a locus which might have two different types of resistances, so a dual type resistance. So to put this in pictures, this is what usually happens with a race-specific resistance. There's a RLM9 gene, it interacts with an effector, AVR-RLM9, but it also interacts with this oligogalacturonides. So it interacts with two different proteins, and it creates a 5% resistance effect, a quantitative resistance effect. At the bottom, you see programmed cell death with race-specific, and on the right, you see many different other pathways. And this is partly overlapping, so the picture is getting a little bit blurry now. We cannot really say these are totally different types of resistance reaction. It has been described before that loci controlling quantitative resistance contains some of the same genes, but quantitative resistance goes far beyond this. However, they talked about loci. Loci means a region, because they didn't want to say, they had no examples that the same genes actually do both. They were thinking that there are 5 or 6 different genes in there. So we showed here that one gene can do both. And coming from the gene for gene hypothesis with flaw, we should maybe modify it a little bit and say it's a gene for multiple gene hypothesis, which we have here. So implications for breeding would be that all the R genes we have in our cultivars are worthwhile tracking with markers and incorporating into new cultivars. They're not useless if the qualitative resistance is lost. Also, we know now that this one locus, RLM479, has 3 different variants, and maybe we can also stack them and increase in one cultivar and increase quantitative resistance. So there were a lot of people involved here. Paul Vollrath was a PhD student who did most of the studies. A lot of people from Justus Liebig University, also from JAST, Julian Gabor, who helped with data analysis, and some commercial partners, breeders, and our collaboration partners in Göttingen, who did all the fungal characterization and phenotyping. With this, I want to thank you for your attention. Some of the people are marked here. Rod Snowden was here last year. Julian, there's me, then Julian, and then Hamid Shavla. This is an old picture. There are many new students which were involved in this project. Thank you for your attention.
Thank you, Professor Obermeier. If you have a question or analyzing this marvelous presentation, please.
Thank you. Thank you very much. Great talk, Christian. I'm familiar with the results, as you said. But I have a curiosity. You talked about qualitative and quantitative disease resistance.
Yeah.
We know that a practical breeder is able to actually breed for multi-disease resistance using qualitative or race-specific genes. Do you think it would be possible at some point to have cultivars on the market that have quantitative resistance for multiple diseases, like not only 𝘁𝗁, but also sclerotinia or verticillium and oilseed rape?
Yeah, so this should be possible. This is another thing which should be addressed, to breed for multi-disease resistance and finding some of the genes which are involved in resistance to more than one pathogen. And this has been shown repeatedly that resistances to more than one pathogen can be localized in the same genomic regions. And often people think, yeah, maybe there are different genes in there, but there are no examples until now that exactly the same gene is involved in resistance to more than one pathogen, except in Arabidopsis. There are a few examples. So I think we will also find this in crop plants. And with the new technologies nowadays, we can pinpoint this down. We can much more exactly identify the genes involved in that. So multiple disease resistance is a big topic, and I think this will be possible in the next years to find regions like this.
Yes, please.
Herr Obermeier, mein Name ist Johann Bujo. Ich arbeite im Institut für Pflanzenbau in dieser Universität. Meine Frage zu Ihnen ist, welche Wichtigkeit spielt dieser Erreger in Landwirtschaft in Deutschland? In Rumänien, wir haben eine große Fläche mit Winterraps, so ungefähr 500.000, 600.000 Hektar jedes Jahr.
Ja.
Und wir haben auch diese Krankheit in Rumänien.
Yeah, so in Germany, sclerotinia and 𝘁𝗁 are the most important diseases. There's clubroot also, which is now coming up. But in the last years it was not so bad, but we had very bad years with 𝘁𝗁 problems. And breeders are marketing their cultivars now with the label RM7 inside, so it's a very important thing for the farmers to get resistance cultivars. They're also working on this quantitative resistance and to find new R genes to stack them into their cultivars. It's quite important in Germany. There are countries in the world where it's even more important, like Australia. It's devastating because they grow plants now much earlier, and then they have something they call upper canopy infection. So the plants die very, very young, and it's a really severe problem. So also agronomic changes will have an impact on this disease. So we will get more, not less of this.
Thank you.
Yeah.
Thank you very much. I'm sorry, I don't speak German. Great talk. Thank you very much. It's not my topic because I'm a vet and a virologist. Just two small questions for them. We know that there is a lot of plants who are resistant to very high temperature, for example, in the United States in the desert, and that plants are infected with the fungus, different kind of fungus. But the fungus are infected with the viruses.
Yeah.
And this infection, it's actually the quality of the plant to resist to the high temperature. Did you screen the viruses who infect the fungus?
No, we didn't screen the 𝘁𝗁 races if they're infected with a fungus. This is your question,
Yes,
No, we didn't do this, and there's no reports for this fungus of any virus infection. But we know there are other diseases where, for example, rhizomania of sugar beet, where soil-borne fungus is transmitting a virus, making the real damage, the hairy root system. So in some systems, this is very common, but nobody found a virus in 𝘁𝗁 until now.
Thank you very much. And the second one, did you apply the CRISPR-Cas9 technology in your case?
Yeah, we are working a little bit on this. So we applied this. We got a construct from some collaborators in the UK for a gene which is in one, that's for a different disease, but which inside a QTL, which is involved in hormone signaling. And we knocked this out, and we are just testing this. So we're working a little bit with CRISPR-Cas. There's 𝘁_ċi who will give a talk tomorrow on CRISPR-Cas editing, not for disease resistance, but for other topics.
Dankeschön.
Yeah, thank you.
You said they don't know German. It's anyone who want to put a question to our colleague? No? If not, thank you, Professor Obermayr, for your excellent presentation. We will continue with the second plenary presentation, titled Innovative Strategies to Enhance Yield and Quality of Vegetables Under Sustainable Perspectives. Presenter is Professor Gianluca Caruso from University of Naples, Federico Duie di Napoli. Please, Professor.
Good morning to no. Good morning to everybody. I've come again after pandemic to give a presentation, so I'm very happy with this. And I'm giving a presentation regarding the innovative strategies connected to vegetable management. But I've agreed with the co-authors, Professor Vasile Munteanu and the others, to focus on rocket because of the short time, of course, available for the presentation. So, but the strategies I'm going to show are common to most of the vegetable species. So we worked a lot on rocket, and we've chosen this species because we also have 2 PhD students, one at this university, the other one in Naples, so that's why we also have a stronger cooperation with this species. Well, we published some papers in the last years. The first one was a long time ago, but in the last years, we have focused a lot on this species and published a number of papers on good international journals. This is a view of the plant in object, and you don't know the flowers because it's mandatory to harvest this plant prior to the flowering phase, and that's why, but the flowers are yellow. Maybe you all have seen these flowers. And just a note that it has a Mediterranean origin, but currently it's spread everywhere worldwide. Because with the plant breeding, there are varieties adapted to all the environments. So it's not difficult, and indeed, Professor Solero and his team have been growing up these species for 5-6 years. And Italy, particularly 𝘁𝗱𝗆𝗆, is the core of European production. That's almost 70% of European production is achieved in the 𝘁𝗱𝗆𝗆 in Salerno province, that's in Italy. But of course we expect that in the near future other countries, Romania included, will contribute to the production in a higher extent. So this is just a look at the main way to produce baby leaves. The left-hand side slide picture, and on the right-hand side, the two major market proposals, that's in bag and in vessel. Mixture is also spread, but not so much. That's most of consumers prefer to buy just rocket, and in case to mix it at home with other salads. Referring to circular economy, in the case of rocket, we have a different circuit. That's not the common one to valorize the residues, because the residues are in small amounts in the case of rocket, and as this species belongs to brassicacee, which are known to have a good impact on soil disinfection, that's the biofumigation. So the few residues are worth to be plowed into soil to give this beneficial effect. Rocket has become a major vegetable inside the perspective of Mediterranean diet. In addition to the last millennium technological innovations, very, very big and impacting, we have proposed and we've been managing other strategies, innovative strategies, and we've already published something, as we saw in the previous slide, regarding these strategies. But we've been carrying on with other research because it's important and needed to deepen all of these strategies, starting from the crop system and cycle, the cultivar adapted to each environment, and we'll see that there is a major issue concerning the nitrate concentration. It's an actual issue indeed. The European regulation has included values, official values, thresholds not to overcome, because there are some concerns with human being health, and we'll see in the next slide some of the values. But commercially speaking, the big companies, commercial companies, have lowered this threshold because have understood that consumers expect a lower nitrate concentration, because currently it's a common knowledge that nitrate, in the end, we'll see shortly the path, in the end can damage the human being. So fertilization, irrigation, biostimulation, biofortification are very, very modern technologies to enhance yield and quality, always in the perspective of sustainable crop management. And then there is also processing and storage technique, which is the second part of the chain, which have been developing very, very fast, proposing to the market every year new items. Well, why we speak also of yield and not only of quality as in the last decades, because we know that we expect that the population is going to increase. So, but the lengths worldwide tend to worsen. So that's why we need to have to get higher yields per unit of surface. We have, as we all know, different crop systems. The organic was a target, but currently the actual target is biodynamic. Indeed, the biggest companies have converted to biodynamic in the last decade. Maybe officially this matter isn't well known because the certification is still organic certification. So there isn't a biodynamic certification, but the European Union reference is the same for both crop systems. Then there is hydroponic, and rocket is also grown by hydroponics. This is a model of the rocket crop systems in 𝘁plain. There are big greenhouses, polytunnels, and this is a system with multiple rows and very high densities. But one concern extended to all vegetable systems is the biodegradable materials. That's the goals are biodegradable, not only mulches, which is still, existing, but also to cover the greenhouses, because it's a big issue to dump all this plastic material. So we've been working always in this field. Then the nets, which have been replacing the paints, because the nets last some years. The paints every year are removed in autumn by the rain, and have an impact on soil and on the waters. This is an example of a solid system. This is a float system. That's all the trays float on the nutrient solution. And these are some chances to choose an alternative to the traditional chemical fumigation. Indeed, we know that some products have been banned, and so it's mandatory to search for new solutions. One is solarization. We published something on this disinfection tool, and it's very environmentally friendly. Then beneficial microorganisms, very modern tool 𝘁cie, not only for the disinfection, but also as by simulation, as we'll see in the next slide. Then vapor pyro disinfection by fumigation through 𝘁𝗂𝗏 family. And biological control, which is mandatory inside the more sustainable systems such as organic and biodynamics, but the integrating systems, that's the way between the conventional and organic biodynamic, is the most spread system that's tended to meet the commercial companies' requirement. That's currently the target is no residues, no nitrate in the case of rocket. Of course, it's very difficult to reach, to achieve this target, but the tendency is towards these goals. The crop cycles, the major farms in 𝘁plain, which is the core of European production, grow up this crop all the year round. But the major crop cycle is from autumn to spring, and that's just I mean, the more dangerous in terms of nitrate accumulation crop cycle. That's why I highlighted with green color summer to autumn, which is the best one as a compromise between residues and yield. But of course, currently it's not a target for the farms because the farms working with rocket are specialized on these crops. So of course they don't intend to give up some cycles, but they want to produce all the year round. And also the density currently is very high, but the goal would be 1,600 plants, but currently it's about 2,000 and over. And this means also problems with parasites and for humidity. Well, this is one of the papers we published on this crop. And in this case, we worked with the rocket crop system targeting to the fresh market. And indeed, we see that we've written about the mulch. Mulch is not used at all in 95% of cases. That's when the production is addressed to baby leaf production. It's bare soil. But in this case, with this 5%, but very important as well, we saw that biodegradable mulch is very worth and a great compromise between yield and quantity. And this is an example of our trials. As for production, as I said, the major goals are low nitrate concentration. If it's 0, would be great, but currently it's not possible. No antiparasite residues. This is also difficult. Then we don't have, in addition to the traditional quality parameters, we don't have some other parameters except for two very important glucosinolates, which characterize all the species belonging to Brassicaceae family. And they are very beneficial because through the hydrolysis of these compounds, isocyanates are formed, and they are bioactive molecules, both for human organisms and for the soil to control the soil microorganisms. And the shape is very important, as well as the color of the leaves. This is just an example of the glucosinolate molecule, and it's important to focus on the sulfur presence because it's a reference for managing the crop system. That we have to enhance the sulfur concentration in the soil and soil solution, because with the sulfur deficit, we give up a part of these very important beneficial molecules. That this is the way, very shortly, the nitrates are dangerous and in the end toxic to human organisms. That's not just nitrate by themselves, but the nitrate is the path leading to nitrosamines and to methhemoglobin, which have damaging effects on human organism. And we see in this picture that we need nitrate, that nitrogen, which is mostly in forms of nitrate, to get high yield. But we don't need it, referring to the nitrate concentration. So it's not easy, but we have to target the proper compromise between yield and nitrate concentration. Nitrate, as I said, is mostly the form they are absorbed by plant roots, but in some cases, for example, in Amaryllidaceae species, onion, garlic, leek, ammonium is the prevalent form. So this is the way nitrates are important and are reduced inside the plants. And it's important the function of a nitrate reductase enzyme complex. That's this complex work mainly based on the light intensity, as we'll see. Now we have the threshold recommended. It's not an imposition, but it's a recommendation. But it's becoming an imposition even stricter, made by the commercial companies, as I said, that the commercial company in the best way allow for half concentration. So if the product, the big companies like Tesco, for example, if they check, and they do, the product, sampling all the amounts getting to a supermarket. So if the threshold overcome half concentration, as we see this picture, the product is waste. So there are just some examples of the work of a nitrate reductase enzyme complex system. That's the higher light intensity, the lower nitrate concentrations in the leaves. And this is the way to possibly act. Indeed, also the daytime, in the central hours of the day, the nitrate concentration is lower, so it would be more convenient to harvest that way, but there is a counterindication. I mean, if you harvest in the central hours of the day, the turgor of the cells decreases. And as the concentration of nitrate is measured per fresh weight, so the turgor is very important to maintain the low nitrate concentration. So if we harvest in the central hours, which are more convenient, we must, focus on the cell turgor. And this is an example of another tool we've been managing, which is the shading. As you can see, this is a demonstration greenhouse, but we've also managed research. Going from the left-hand side with no shading to the central tunnel with 50% shading to 80%. You see that 80% of the plants grow up very slowly, but a moderate shading is worth to keep the yield high, and in midsummer is very beneficial also for the quality. So these are the results of our research. As you can see in the violet trend, violet line, you see that with moderate shading, the yield increase. But in the other case, that's in winter and spring, even 50% shading is compromising. That reduces the yield. Yeah, we can go on. Irrigation management, I said, is very important, and we've been working also on this topic, because if the cell turgor decreases, nitrate concentration increases. Fertigation, just to show you that the requirements of this crop are in the sense of higher requirement for potassium, lower by for nitrogen. But as you see, the common management is based on the prevalence of nitrate, compared, that's fertigation, through fertigation, compared to potassium. And that's why nitrate concentration tend to get higher. That's the problem is that, of course, a farmer targets the yield mainly, and they don't care a lot of nitrates, about nitrates, unless the commercial company force them to keep low the nitrate concentration. So that's why, contrary to the plant requirement, usually nitrate prevails on potassium. So this is an example of how yield and nitrates accumulation don't agree to each other. That you see that we have with some mode of fertilization management, we have higher yield but also higher nitrate accumulation. That's a particular very important for rocket management is that even non-fertilized control accumulate a lot of nitrates. That's nitrate rockets is considered a hyperaccumulator of nitrates. Even with no contribution, no supply of nitrogen, usually nitrate form, there is an accumulation, you see, that's almost 3,000 ppm of nitrate with no nitrogen fertilization. So by simulation and biofortificants, we know that among the biostimulants there are the microbic types, non-microbic types. Among the microbic types, we all know the arbuscular mycorrhizal fungi and trichoderma. We can't use arbuscular mycorrhizal fungi in rocket because brassicaceae don't accept this mutualistic relationship, that this symbiosis. So trichoderma is very used on rocket. Then the biofortificants have got very spread currently. That's mainly selenium, which also works as an antioxidant. Then iodine is important for people who have some problems with the thyroid. And silicon is also a good and beneficial biostimulant. And they work increasing, enhancing all the major characteristics of plant management. We worked also on microbic trichoderma and non-microbic compounds, also combining them. That's the combination hasn't led to benefits in terms of yield and quality. So choosing a microbic trichoderma or a non-microbic is the same, has the same effects. But trichoderma also has some protection effects. That's in terms of parasite, fungi, soil, insects. So trichoderma is preferable from some point of view to the protein hydrolysis, for example. Microalgae is a very, very modern current tool for biosimulation of plants, but we also eat some formulations of microalgae that they are sold in all the specialized shops, and they are very useful also for the system plant soil. Currently, there are some producing systems, very, very modern, which are the photobioreactors. And in Italy, some companies, some firms have been creating these systems, just working on production of microalgae, which are more and more required by consumers. This is an example of a modern bioreactor systems. You see that different species give different colors to the view. There are different genus, for example, Arthrospira chlorella. Several effects depending on several factors. Harvest is very important. Also refer to the second phase of the chain, that's the post-harvest management. That's, as I said, there are some crop cycles which are better in terms of yield, other in terms of quality. But it's important that harvesting the rocket is referred to maintain a temperature, a cold temperature, since harvest to the industry management, transformation, processing. That's 4 to 8 degrees. Initially, the temperature levels was 0 to 4. Then as it's difficult for the operators to work at very low temperature, it's been increased, but it's a fridge temperature. So just after harvesting, the product is put inside this fridge, which are close to the farm, and transferred immediately to the industry to produce baby leaf packaging. And we can see also 𝘀𝗶𝗲𝗀𝗃 on the right-hand side when he spent 2 months at our department, and we visited a big farm, about 100 hectares, producing just rocket, and this is the mechanical harvest of the product addressed to the baby leaf production. Then, when it's addressed in 5% of the cases to fresh market, is harvested manually. It's important the length of pishul, because pishul accumulate a lot of nitrate. So the pishul shouldn't exceed a certain length, possibly. And also, we don't have to target yield with exceeding with nitrogen, mainly nitrate supply. It's best to get a compromise between yield and quality, mainly referred to the residues, nitrate and antiparasites. So an innovative tool is, as maybe most of us know, the modified atmosphere, which is indeed targeted by companies producing baby leaves. That modified atmosphere means that we lower the oxygen concentration inside the package, and this way we reduce the oxidization of the product, and at the same time we take the carbohydrate, the CO2 concentration to a level which is about 100 higher than the level we have in the air we breathe. In conclusion, we have seen a lot of strategies, innovative strategies, which help us to get a product which is indeed required by consumers, and that both the farmers and the commercial companies are interacting to provide a product which is, of course, satisfying for the farmers in terms of yield, because the farmers want to get yield to get money. And the commercial companies which have their own targets, threshold, referred mainly to nitrate concentration, and consumers who, of course, don't care about the yield, but care about the quality, mainly referred to the benefits to human organisms. Thanks a lot for the attention.
Thank you, Professor Caruso. In Romania, Diplotaxis tenuicola is rucola. Maybe you know.
Yeah.
Yes. We have a question for a colleague.
For me.
Hi, Professor. Congratulations for your very interesting paper. If your paper was about rocket, I hope the next one will be about meat, to have a circle. I have a curiosity.
Or pizza. Pizza with rocket.
Yes. I have a curiosity. Please say something about the market of the rucola, total yield, value, and where is valorized the rocket? In domestic market, what is exported? Thank you.
Yes. About the market, we have, as I said, that 70% of production at national level is located in Salerno province, Sele Plain, which is one of the most important plains in Europe, very technologically advanced. And the production is addressed to all the Italian territory, but also all Europe, because going to Scandinavia, the production is more and more difficult currently, in spite of the efforts of breeders to get new varieties adapted, as we saw with Professor Vasile Solero, that's here in greenhouse, we say, because in Italy, just in summer, we also grow the big farms, can grow also rocket, uncovering some tunnels, and so devoting a part of the surface to the rocket. But in summer, the product is more difficult even for the commercial companies, because it's more difficult to prevent flowering phase, to get a good quality in terms of sides, leaf sides, cell turgor, color, and even more difficult, even harder for the commercial companies to manage this product and to transfer it all Europe around. So the big companies, I gave you the example of Tesco, are very involved in this business because has been an increase in business for the last 2-3 decades. And so we don't know if it lasts, or maybe there will be another innovative products covering the market spaces actually belonging to rocket, because rocket is the queen of the baby leaves currently. There are also other species we know, lettuce, spinach, valerianella, but rocket remains the leader in this field.
Thank you. Professor, tell us if hydroponic cultivation and biostimulator in hydroponic cultivation is more efficiently comparing with cultivars in soil of this rocket?
Sorry, you mean that biostimulants in soil or soilless?
Where are more efficient, in soil or in hydroponic?
Well, in soil are more efficient because the hydroponic system is made in a very efficient way. That's all. The nutrient management is very efficient, very precise, calculated prior to the managing, using all the technology available. So the plants usually have all they need in terms of the 13 essential nutrients. We know that there are 6 essential macronutrients and 7 essential micronutrients, and the plants have everything in soil systems. In soil, of course, they could also have potentially the nutrients available in the soil, but there could be mainly, for example, in Mediterranean areas in summer, there aren't always the conditions for the optimal absorption because of temperature overcoming 30 degrees easily in July, August. So in that case, the biostimulation mainly achieved through spraying the aerial part of plants. So it's easily all the products which are, it's a mix of products, organic compounds, phytoregulators, nutrients, and they are immediately absorbed when supplied, when sprayed on the upper part. Because the problem in difficult condition, mainly low temperature and high temperature, is in with soil absorption, so with the root working. So biostimulator, the biostimulation is both with non-microbic and microbic, but mainly with non-microbic is much more efficient when working in soil.
Okay, thank you. Is anyone want to address a question? Please, no. Please, Professor Muntean.
Thank you. Thank you very much. Thank you for your presentation. Congratulations. When I hear sustainability, it's like a challenge for me because many people confuse sustainability with durability. For instance, Eiffel Tower is durable, but it's not sustainable. But rocket is sustainable because if we cut the leaves, in the next year, plant have the possibility to renew.
Regrowth, yes.
So when people had the word sustainability, become nervous. Oh, sustainable, what is this? Again, sustainable. This is very important that in your work and our work, you have present some solution to be sustainable. This is very, very, very important because organic agriculture is clear, is good, is friendly, is okay, but it's difficult to feed more than 7 trillions of the people with organic agriculture. It's not efficient, economically efficient.
Yes, sorry.
Yes, this was my intervention.
Yeah.
Thank you.
Well, I've spoken generally about the overall sustainability. Of course, we know that sometimes referring to environmental sustainability, but we all know that the sustainability is a very broad concept, including technical sustainability, environmental sustainability, social sustainability, economic sustainability. So all these aspects of sustainability make the sustainability, and one or 2, 3 aspects can prevail on the others, depending on the context. So, but environmental sustainability is, in our case, you are a coder, in our case, is the major target. So, because we've spoken about mainly technical sustainability and impacting on the environment. So that's a major target and actual very modern target because more and more consumers have been becoming sensitive to the quality of the product. Indeed, some products also have on label the percentage or the presence of antioxidants. So we have in the more advanced countries a small percentage, maybe 5%, of consumers focusing on all the quality characteristics of the product. So primary metabolites, secondary metabolites, small percentage, still small percentage, but increasing.
Thank you. There are questions? If not, thank you very much, Professor.
Thank you. Thank you to you. Thank you for your attention, for your patience.
Next presentation will be titled Modern Trends in Food Contamination Analysis, an indicative research proposal on mineral oil hydrocarbons, migration potential from food contact material into food products. Presenter is Dr. Engineer Madalina Matei from Animal and Food Sciences Faculty. Please.
Hello, dear guests. I'm very happy to be here today to talk about food contamination. And I want to share with you an interesting story about mineral oil hydrocarbons. The story begins, I will say, recently in 2008, when mineral oil presence on the food chain really became a problem because of a critical case of contamination reported in Ukraine for some sunflower oil batches. In Romania, mineral oil contamination is a new topic which is little explored. In countries like Italy, Germany, the Netherlands, or maybe Belgium, we can find the latest trends about analyzing methods, toxicology, or different legislative frameworks. Mineral oil hydrocarbons are a group of modern contaminants from petrogenic processes which contain some proportion of saturated or aromatic hydrocarbons. The contamination with mineral oil in foods comes from a variety of sources. For consumers, the most important sources of contamination is via packaged food, because of approved uses of mineral oil as processing aids or in some additives in food contact material. And we have some examples: the paperboard packaging, the printing inks applied directly to the packaging, and also the paraffinic mineral oil used for waxed food paper. For checking potential food contamination via packaging, we propose a preliminary research regarding the migration of contaminants from food contact material into products by testing mineral oil hydrocarbons in product and in contact material to evaluate the contamination profile and finally to establish some future demands for develop an extensive research on this area. This study calls firstly on producers on the supply chain because of the variability of sources of contamination and also the requirements needs on processing chain, especially to the packaging step. Because data on occurrence of mineral oil contamination is currently available only for a limited number of food, this study is addressing to all authorities at national and European level, and not least for the consumer. It is important to know that 𝘁𝗏 topics focus primarily on food and consumer safety. Current state of art, or something about the toxicology and legislative background on this topic. First, 𝘁𝗏 has different toxicological potential. We knew until now that MOSH can be easily assimilated in the body and accumulated in fatty tissue. Now, the toxicological relevance of the effects shows for MOSH in rats is questionable for humans. In the latest studies, it is likely that the present dietary of exposure to MOA doesn't raise concern for human health. For MOA, genotoxic and mutagenic nature was demonstrated. In the latest study, additional data on toxicity and exposure to 3 or more aromatic MOA are the main subject for the risk assessment. For example, is likely almost 100% for toddlers and more than 66% for other group age that the dietary exposure to MOA can be associated with DNA damage, potentially causing cancer or raising some important concern on human health. No toxicological data values are available for evaluation so far, and no tolerable dose are available. All these opinions are shared by different authorities such as EFSA, BFR, or ANSES, who believes that the priority should be given by reducing the food contamination, and it recommends limiting consumer exposure, especially to MOA. If regarding the legislative background, there are currently no legal limit and no European regulation for the presence of MOSH and MOA in food. There are only some benchmark levels proposed over the year, but never officially entered into force. Only some minimum performance criteria of the method applied of 𝘁𝗏 analysis are available so far. The GRC technical report in frame of European Recommendation 84 by 270 is a real guide for sampling, data analysis, and data reporting for mineral oil hydrocarbon in food and food contact material. The Standing Committee on Plants, Animals, and Food from European Commission is also an important framework because it presents the condition about the withdrawal or the recall of products from the market in case of contamination. EFSA has carried out recently a scientific opinion, underwent a public consultation from March to April this year, and now this scientific opinion with all the updates are available on EFSA journal. 1 month ago, in September, he appeared on EFSA journal. For the assessment of mineral oil hydrocarbon, EFSA completed a comprehensive review of the scientific literature, including data about the toxicity especially. EFSA regularly collected information about the exposure and the levels of 𝘁𝗏 in foods from European member states and also other stakeholders. This data being collected between 211 and 221. It is an important change. We hope that will enter into force as a regulation. Knowing all of this, we wondered if mineral oil hydrocarbon can migrate from food contact into food. We will see if this preliminary study can be used to evaluate the situation of contamination and then to develop an extensive research on this area. For answering to all these questions, we carried out a test to see what is the migration potential of mineral oil hydrocarbons to food via packaging. So it was carried out a 𝘁𝗏 content analysis performed based on 4 samples of milk. No processing or packaging processes was applied to milk samples before starting the test. The 4 samples were divided into categories: control samples represented by milk, test samples, and food contact material represented by bleached waxed food paper. Because under the temperature influence, mineral oil can be able to volatilize. For testing migration potential, some samples of liquid milk was covered with waxed food paper and lyophilizated for obtaining freeze-dried milk for testing. Mineral oil hydrocarbon analysis is using 𝘁cie feed technique based on an actual protocol who meets the analytical performance criteria mentioned in GRC guideline. The 8PLC coupled to a GC was set up with a dual channel so that 𝘁𝗏 can be analyzed in the same time. For collecting and processing data, the 𝘁𝗏 software from Thermo Fisher Scientific can be used with good results. The analysis protocol for mineral oil is particularly demanding, especially for food, including several work steps. Among the most important, we list microwave-assisted saponification or epoxidation necessary to eliminate the interferences and achieve sensitivity. Being many work steps beyond all is important that to obtain good results is important that the work protocol to be well respected. Let's see what's obtained so far. Different 𝘁𝗏 concentration was measured in control samples with some important differences between the samples. After 7 days of lyophilization, an important increase of MOSH and MOA content was revealed for the test samples. For freeze-dried milk, the amount of mineral oil found was considered alarming, and it indicates an important sign of contamination. For sure, the MOSH and MOA concentration, the levels are important for us, but we especially want to discover from where it comes the differences between control samples and test samples. If we're looking at food contact material, we will see also an important contamination with MOSH and MOA. The same result indicated that there are large differences in terms of proportion for MOSH and MOA between control samples, test samples, and contact material. Especially was highlighted an important increase for MOA fraction, which indicates a critical situation. To establish the migration potential of mineral oil was presented the chromatographic profile of the milk samples before and after the lyophilization. And we have two examples of chromatograms before and after the lyophilization. As we see, no humps or some little humps are observed for the control samples, while in the test samples, it indicated an important hydrocarbon hump, especially ranging from 16 to 35 carbon. Control samples and test samples chromatogram in relation to wax paper chromatogram show a similar MOSH distribution. The two green symbols indicate the migration potential from waxed paper to milk. The same rotation of MOSH can demonstrate that the migration of contaminants from contact material in food appears to be realistic and may be due to the volatilization of paraffinic wax used in paper making. We talked briefly about food contamination of food through packaging. We conclude this preliminary study saying that mineral oil contamination of food through packaging is still debatable. What I would like you to know is that the practical use of specific packaging is not enough to prevent contamination because the probability of contaminant transfer is an important warning signal if some similar waxed packaging are used for packaging the foods. Special attention, according to the literature, must be paid to the packaging like pizza boxes, sandwich paper boxes, or bakery paper bags. In generally, the packaging used for hot products. The future demands goes sure for all the manufacturers and private retails, but also to all authorities at national and European level, especially to establish some requirements for mineral oil hydrocarbon in the European legislation. As I say initially, in Italy there are many concerns about the mineral oil hydrocarbons. So this study was a collaboration between IASI University of Life Sciences and the University of Udine from Italy. And I had the opportunity to work with the chemistry group for big companies, I would say, in Italy regarding this subject, like Barilla Pasta, Ice Cream San Montana, or Perugina Chocolate. And I want to thank them for this beautiful collaboration. Also, I thank you for your attention, and I hope you enjoy my presentation, and I want to say you to stay tuned to see the future challenges. Thank you.
Thank you, Madalina. Any questions? If
Nobody wants to eat pizza anymore.
No. Thank you very much for your presentation. The last but not least presentation, titled The Science of Science Communication, it's presented by Dr. 𝗟𝗶𝗳𝗲𝗀, belonging both to Sorbonne University in St. Paris and on the Department of Public Health, Iași University of Life Sciences.
Hello, everyone. Thank you very much for this invitation. The last one, so every time a talk before brunch or lunch, it's a big challenge, right? I will be, don't worry, the brunch, it's after me. The science of science communication. Why science of science communication? Because science communication is a science, because we already teach the science of communication to our students in a lot of universities across over the world. And our students will be the future specialists, whatever the field, and our students will be the future PhD students, so the future maybe researcher and teachers. And they have to know how to communicate their science with the public. And this is the reason that it's my topic, science of science communication. The term science is a vague word, and for a huge category of information. And this is the truth. We like to say that science, it's a hard science and a soft science. Hard science, for example, the subjects that are included in this category are physics, mathematics, chemistry, biochemistry, biology, anatomy, astronomy, just to name a few of them. And soft science included fields to study like sociology, psychology, political science, anthropology, and again, just to name a few of them. The COVID-19, the pandemic period, and unfortunately, I like to say the infodemic, because in that period, everyone became a specialist in vaccines, in immunology, and in virology, whatever, epidemiology. But infodemic surrounded it have driven the point home once again on many, many issues. And on timely issues like climate change, which is true, gene editing, it's scary for a lot of people, for the society, our society. Artificial intelligence, that was presented several weeks ago by our rector here. And individual nutrition, talk before, after me. Healthcare. So in all these things, the public, the large public, our society, expects researchers to speak up and present their research. The science, in my opinion, is not only my opinion, it's probably the best source for evidence needed to answer to the benefits for humans, animals, and for our society. And public outreach has become an issue of growing importance of science. And in this case, an active communication between the researcher and the society, maybe you, I mean the public that you are today, it's necessary for the scientific community involved in developing science-based policies. However, the trust of the public, of our society, in scientific progress needs to be earned, to be kept, which of course will require inclusive, a self-reflecting, an honest and inspiring science communication. And if our nation, or if the public, are better informed about the scientific progress, whatever the fields, right? They can not only make better decisions personally, but also they can influence decisions that can affect their towns, their nations. How does science communication help bridge the gap between the science and the society? Because unfortunately, there is a big gap between us, like a researcher, and our society. They cannot understand. And if we want to progress, we have to share with the public our research, and we need the support of the public. Otherwise, it doesn't work. In 1976, Edward Hall, it's a very famous anthropologist, American anthropologist, developed an iceberg analogy of culture, which is very nice in terms of communication, science communication. For example, in this iceberg, if the culture of the society was an iceberg, Hall reasoned that there is some aspects visible and some aspects hidden. For example, if I want to share with you my scientific project in virology, and I will present it to you just, it's neuroscience, because our brain can memorize in a few seconds only 10%, which means information like numbers, data, facts. But if I want to share with you more in my scientific project, I have to make sure that I address the 90% that are hidden, which means because you don't know my experience, my feelings, my wishes, my hopes, my health, my future, the past, my language, my culture. So if I want to share, it's 10%, and to be sure that it's 90% to share with the public, all the information. So 3 ideas, don't worry, just a few minutes. A few ideas and several, let's say, tips to be a good communicator when you want to share your science, again, it doesn't matter the fields, mathematics, physics, agriculture, etc. So first idea, the smartest speak the language of the most uneducated who is supposed to understand them, not the language that proves their own education. In other words, if I want to share with you my scientific project, I have to speak with you in terms of words and in terms of language I'm sure that you can understand my virology subject. Otherwise, it's for nothing. You are scientific, most of you. You have a background, scientific background, so it's easier. But with the large public, it's not so easy. A second idea. Only those who communicate form opinions, and it's really, really true in our life. And another one, do not ever underestimate the basis for some paradigms. If you want to change something, in the real life, we say, the people doesn't change. They cannot change. And if you want to change a paradigm, for example, I don't want to be vaccinated. I can understand. But if you want to change this, you need a very good strategy in terms of science communication. And I like this Chinese proverb in terms of strategies. When the wind of change blows, some builds walls and some others windmills. And this is true. Two different strategies. Well, science communication. What? If you want to share your scientific project, you have to start with something that you feel okay. Some of us can easily speak with the kids, with the children. Some of us, so it's easier to write an article. Oral presentations, like I do now. I was forced, but I liked. Social media, some of us, they like to be on the television, radio, etc. But anyway, the last message, the goal is to share your scientific project. So you can choose something that you feel okay. How to launch your science communication? I like this expression. We hear a lot in London. It's not rocket science. which means, do you know this? Of course I know, it's not very complicated, it's not rocket science. So really, it's not rocket science. And these are the 6 tips for science communication. First one, know your audience. Very important. Think about your message. What kind of message I want to send to the society? Tell a story, not complicated. Avoid, please, the jargon. Nobody understands nothing about your science because it's too complicated for them, maybe. Make it relatable, I mean your story. And don't forget the visual. 6 rules, 6 tips for science communication. First one, know your audience. Why you are here? Why do they listen to me? And what do I want them to remember when home? Very important. Second one, think about your message. And the message is all the time first. Don't say, please, your project, my project is too complicated to be explained easily, because it's not true. You can do it, you can explain easily, if you want. Tell a story. Be personal. Avoid, please, the listing discoveries. Why? Because it's so boring. Show your passion. This should be very easy for you. Why? Because you are a researcher, you work with passion, you like the researcher, so show your passion of your scientific project. And of course, create a narrative in your story. Avoid the jargon. There is a lot of papers. And we know, so non-scientists feel shut out and confused by articles that use technical language, even if it's definite. We cannot understand all the technical. How can you avoid the jargon? Simple, it's for free. Just Google. Google de-jargonize. So this is 2 phrases that I enter it's a topic in virology, dogs, etc., complicated. All the words in red, for a large public, without any scientific background, they cannot understand your message. So you have to move out, or these technical words. This is not for the topic, but I think it's important for you because in front of me, I know my public. I know who are you, because it's easier. It's my university, right? So this is because we want to publish, right? And we need a lot of citations. So this is very interesting because in terms of, according to the 2 researchers, they analyze the jargon in more than 21,000 scientific manuscripts. The result is papers with a high fraction of jargon, they receive fewer citations. So even in science, a lot of jargon, few citations. Interesting, right? Another rule, make it your scientific project relatable. And if you work with some crazy numbers, like I do in virology, size or distance, bring it back to normal everyday stuff. This is a slide for my students, and was confused because I said, okay, the number of the viruses on the water, oceans, is something that's 10 to 30. How many is 0, they asked me? It's a lot, it's a big one. And if I said, okay, this virus is only one line, and this one line, it's more than 100 million light years into the space. This is the number of the viruses in the oceans. Okay, it's a lot. It's easier to understand the numbers, the size. And in the end, it's not just about the words. According to this theory, my words today was only 7%. I don't know if my voice was okay. I don't know if my hands was okay. I try to not use casual things like this one. Why? Because it's a neuroscience. We hear with our eyes. So if I do something more with my hand, with my body, it's language body, so you don't hear me anymore. Just, we lose a lot. So don't focus only on the words. There is a lot of action how we teach our students for science communication. So there is across Europe, across all the world, a lot of action. One of them invented by two researchers in 2014 in England. It's Pint of Science. And Pint of Science is everywhere in France and everywhere in a lot of, I think it's 22 different countries now. So we spent a very nice evening, listen to a very good band, jazz band, whatever, the music. And we invited researcher. Of course, we don't have, but we drink a couple of beers, and we listen to science. And the public, it's lay person, nobody, so they wanted to listen, and this is great. This one, Tactile, it's action. We learn science to blind people. It's all across over the world. And I like one, Fait de la Science in France, there is since now, I don't know, 35 years. We learn, and we share the communication, the scientific project with the people, with the nations. And I like, my preferred is my thesis in 3 minutes, in 108 seconds. And this is my last slide. And I will wish you, Mr. Rector, to have this, my thesis in 3 minutes in this university, maybe in Romania. It's easy. So what is this? The PhD students have to present their very complicated scientific project in 3 minutes in front of the students, whatever the topics, mathematics, medicine, anatomy, whatever. And the winners will be a second step in national, and after that, a global, and it's great. I choose one student, it's a she. She was the winner 2 years ago from Australia, and she explained a very complicated pathology after the stroke, but this pathology for human affects the communication. Let's say.
Imagine you're dropped off in a foreign country, say Japan, without warning. You only speak English, and everybody around you only speaks Japanese. How would you survive? How would you order a coffee? Have a chat? Make friends? Hold a job? This is the daily reality for people with aphasia. Aphasia is a language impairment often caused by stroke, which is a disruption of blood flow to the brain. It affects a person's ability to speak, write, read, and understand what somebody is saying to varying degrees. This can range from having difficulty finding a word every now and then and understanding almost everything, to being unable to say any words and understanding very little. Now, we've gotten very good at describing different aphasia types based on their language skills, as measured by language tests, such as picture naming or matching a sentence with the right image. Ultimately, though, speech and language therapy aims to improve someone's ability to communicate, to have a conversation. The problem is that we don't really understand the concept of communication very well, nor do we know exactly what kind of skills are required to communicate efficiently. So remember I told you don't speak or understand Japanese? You'd score pretty badly on a test of your Japanese language skills, right? Yet I predict that you'd still be able to order a cup of coffee and have a chat with someone in Japan. Just as we see that some people with a very severe language impairment are still able to communicate quite efficiently. My research aims to find out what enables people with aphasia to communicate despite their inability to rely on their language skills. Now, from research with people without brain damage, we know that in addition to language, there are at least 3 elements that people use when they communicate. 1, we don't just communicate by using language. We use other means to transmit our message, such as gesture, facial expressions, body posture, and intonation, so the way we say our words. 2, communication is interactive. It doesn't just rely on my language skills, but also on those of the person that I'm speaking to, and how they can help me out when I get stuck. 3, communication always takes place in a specific setting or context. So expressing a sudden urge for coffee is easier when you're next to Starbucks compared to when you're on a hike in the woods. By manipulating these 3 elements in the lab, I want to find out if people with aphasia can rely on these elements just like we do in our everyday conversations, or if it's different for them due to their brain damage. By getting a better idea of what communication means, instead of just language skills, this can inform our therapies, our measures of communication, and hopefully help those who struggle to communicate every single day.
Thank you.
Thank you, 𝗟𝗏𝗳𝗲𝗀.
The branch is over. It's open.
Any question, please? Yes, we have one, Professor Gianluca.
Congratulations for your presentation. Just a curiosity. You said, my opinion correctly all the time, that the scientists should endeavor to bridge the gap between science and the common citizen. But in my opinion, and I would like to hear yours, also, we should also stimulate the common citizen to upgrade and to tend and learn more scientific or technical terminology, because sometimes it's difficult. I'm thinking of my presentation, for example, the danger of nitrates. So it's very difficult to explain differently to people. Otherwise, you can say there is a danger in this product, but it's too general. And of course, the citizen should just rely upon you, upon what you say. Differently, if a citizen understands something, even of at scientific level, of course not high level, but basic level, he can make more aware choices, in my opinion. That's I would like to hear yours.
Yes, it's true. I mean, this, for me, we have to educate these people, right? And some, like you said, nitrate salts, yes, because they heard this word a lot on the television, radio, and it's like, memorize in the same words. But I think we can educate it with different technical words, but not all of them. I mean, if I have to talk about the gene editing and CRISPR-Cas9, I'm not sure that you will understand CRISPR-Cas9. Your colleague, that was the first presentation, of course, it's a technology. We use this for humans, animals, and for plants in agriculture. But for the lay person, they cannot understand. But if I can say that with this technology, without any explanation, I can block something, I can name gene, but without intron, without exons. But I can block something, that I can block a protein, that protein, you will be healthy, or you will be something, it's easier to understand. But I understand that's your opinion. So yes, it's a mixed, in my opinion, so we have to avoid the jargon. But if we can educate it, lay people with some technical, yes, it's good also. There is a lot of article in the last 5 years. You can just check yourself. So it's so gap between the research and the society, and the COVID infodemic period doesn't help us, unfortunately. And I think it's important for us. We need the support of the public. It's not political. It's a fact. Thank you.
Thank you. Any question, please? Yes.
It's,
jargon titles are not cited when we are talking about research, scientific papers, okay? What do you think this jargon title do get attract attention? What is the major objective for a researcher to put a jargon title? Because for sure, it will not look very scientific if you use a jargon title. But for sure, you have another objective by using it.
I'm not very sure that I understood that. But anyway, for me, it's first rule, know your public. Who are you? So I know you are more or less scientific person, so with a scientific background, right? So I know it's easier for you to understand if I use a jargon. It depends on the kind of jargon, because you can understand. I understood your topic. I'm a vet, I'm sorry, nobody's perfect. So I have nothing to do with agriculture, but I understood your topic. So my question is, avoid the jargon when you know your public. The public, without scientific background. And of course, between the science in the afternoon, we'll use a lot of jargon, right? But we know, we speak the same language. And my slide with avoid the jargon for scientists, it's a new evaluation. It was interesting for me when I saw that more than 12,000 papers, a lot of jargon, and a few citation. Wow. So we like to have citation, right? So it's two things. Be careful in your scientific jargon, because we are scientists. It's two. I know, it's in a peer review. We have to evaluate it. And be careful with the jargon when it's a lay public. But of course, we can educate it with some jargon, of course. It's correct, Carmen? It's okay? Thank you.
Okay, thank you very much.
Thank you very much.
We are still in time. We retain that individual nutrition is compulsory included in soft science, 𝗟𝗏𝗳𝗏, but it's clearly for us that it's time. So the plenary session is finished. Thank you very much for your attention. In the hall of this splendid auditorium, we have a brunch for everyone who is present in this room. So the brunch is under the program till 1:45, and after this we will follow the program listening in our booklet on 13 sections to each faculty. So have a good brunch. Thank you very much.